Good morning. Welcome to the Montagna lecture. My name is May Mahoney, and one of the privileges of being co-chair of the program committee is that you are involved in the selection and introduction of the Montagna lecture at the annual meeting. This year, we are honored to have Dr. Hensin Tsao. Dr. Tsao is a board-certified dermatologist, a professor of dermatology at Harvard Medical School. Presently, he is the director of the Melanoma Pigmented Lesion Center, the Melanoma Genetics Program, the Skin Cancer Genetics Laboratory in the Wellman Center for Photomedicine. Dr. Tsao has been one of Boston's top doctors since 2010. Beyond patient care, he dedicates much of his time to melanoma research and the education of medical students, dermatology residents, and fellows. Dr. Tsao received his undergraduate degree from Brown University, where he graduated magna cum laude and Phi Beta Kappa in English and biochemistry. He went on to receive both his MD and PhD in biophysics and biochemistry from Columbia University in 1993. Dr. Tsao completed 1 year of clinical training in internal medicine and 3 years of dermatology residency, including 1 year melanoma fellowship at Harvard-affiliated hospitals. He concluded his training with a postdoctoral fellowship in the Division of Oncology. Currently, Dr. Tsao supervises an active cancer genetics laboratory primarily in the area of melanoma. He is the author of more than 150 articles, reviews, abstracts, lectures, textbook chapters, and online media texts. He has also delivered more than 130 lectures throughout the world on melanoma, genetics, and skin disease. He has received numerous awards, including the prestigious Marion Sulzberger Award from the Academy of Dermatology. Amazingly, he still finds time to serve on numerous committees. Please join me to welcome and congratulate Dr. Tsao. Today, he will speak on therapeutic target practice in melanoma.
So it's a great honor to be here, and I want to thank the SID, the selection committee, and I think it's a particular privilege, one that's hard for me to even articulate, of what it's like to be in the audience when I first heard the Montagna lecture, and now to be delivering it. So, a couple things to start. We know that BRAF, MEK, PD-1, they're all well-validated targets. Now, I have to say, many of you were locked down here this week, and you probably didn't hear the buzz about the latest target that's generated a lot of excitement, although the science, I think, is completely unsubstantiated. I'm talking about, all right, new target, and for those of you wondering what's that down there in the corner, I gotta get my lasers. That, of course, is the Brady cannon, which is how we pronounce the peptide in Boston, Brady cannon. Oh, come on, my mama laughed at that. She doesn't even speak English. All right, disclosures. I have no financial disclosures. I've avoided them by avoiding actually any intellectual thought. So we'll start out by talking about this disease, melanoma. And if you were to sort of dig deeper into melanoma at a cellular level, you'd see that we started with a melanocyte, shown here in blue. A mutation sort of occurs, and often this is one of the oncogenic sort of lesions, and this stimulates the cell to proliferate temporarily. Additional mutations occur, probably through ultraviolet radiation, that drives this tumor to continue to grow. Over time, cancers adopt the organoid sort of process where there's the microenvironment, there are fibroblasts coming in, and then there's neoangiogenesis with blood flowing through it. At times, one of these cells could sneak into the bloodstream, take off, and produce a metastasis. We also know with the advent of blood vessels, you have circulating lymphocytes going through the tumor, and one of these will eventually recognize the tumor and create an eradication or editing of those cells. Now, Arlene Sharp will be speaking on the immune system afterwards, and she's going to fill you in on a lot of those details. So there's really two areas of targeting that we think about in terms of cancer. We think about immune targeting, and we think about growth targeting. So I'm going to really focus on growth targeting. That's my area of interest. And if we move to the single cell and able to sort of blow this up systematically and strip away the cell and see what's in it, what we find is that on the cell surface are a set of receptors that obviously receive growth factor signaling. Receptor tyrosine kinases become activated in melanoma, and we know that approximately 40% of acral and mucosal melanomas have KIT mutations, ERB4 is mutated, and more recently, Spitzoid melanomas have been shown to have translocations in some of these RTKs. NRAS is also mutated in about 25% of melanomas. NF1, which downregulates NRAS, is mutated in about 16%. And with RAS, there's one pathway going down the MAP kinase pathway. I'm going to stand here a little bit. That activates eventually the machinery to increase proliferation. P16 usually tries to put a brake on that, but it's defective in a large number of melanomas. It's been said to be deleted or mutated in about 60%. CDK4, cyclin D, a partner that drives that cell cycle, is amplified or mutated in a certain number of melanomas that we know about. Of course, BRAF is the big player. About 50–60% of the common types of melanomas have an activating mutation in BRAF. GNAC is a little bit more of a recent addition. It's a small G protein. About half of ocular melanoma show mutations in GNAC. NRAS is another pathway down the lipid signaling, PI3 kinase, as you know, and that leads to AKT. P10 is mutated in about 10%, deleting about a quarter. PI3 kinase, much less, but it's activated about 5% with mutations. That drives AKT to go up, which often stimulates survival instincts through BCL2. MITF is amplified in about 4% of melanomas. All of this conspires to increase the survival urge of melanoma. So, on the one hand, you have proliferation going through the MAP kinase. On another, you have all these secondary mechanisms to help sustain survival. Now, melanoma sits on the skin, obviously, and undergoes a lot of mutations in the DNA. P53, which is the guardian of the genome, is mutated in about 20%. In other cases, it's not mutated, but inactivated by MDM2. We know that apoptosis is lost when P53 is diminished. We also know that inherited deficiencies in DNA repair can lead to melanoma, so clearly there's a homeostatic mechanism for DNA repair. More recently, TERT amplifications and promoter variants have been described in melanoma, about 30% of the tumors, and in fact, in a couple families around the world prone to melanoma. So we know that maintenance of the telomere structure is also an important part of melanoma. So when we start looking at the central core engine, or the axis that sort of drives this tumor, there have been a lot of efforts already in the pipeline trying to target these receptor tyrosine kinases. NRAS has been looked at exhaustively, although it's been very difficult to target. BRAF, MEK, ERK is out there as a new target for testing, cyclin-dependent kinases, and PI3 kinases, and certainly AKT has been also targeted. So within the sort of, again, core engine of cancer and melanoma, there are a lot of exciting new drugs coming down the pike. So for the rest of this talk, in the first part, I'm going to talk really about the two main clinical targets that we have right now, MEK and BRAF, and then I'm going to actually talk about the rest of the session, use the rest of the session to talk about some of the targeting strategies we've been working on in the lab. So let's talk about BRAF. Really, in 2002, this paper came out from the Sanger Center showing that the V600E, and back then it was known as V599, I guess it aged a little bit over time, it was defective in 60% of melanomas, and this is the first mutation in BRAF described in melanoma. By the time we reached 2008 or so, we had a specific agent, the Plex agent, that appeared to show some level of selectivity for the genotype. And this was exciting because this suggested there was a magic bullet approach to the tumor. And by 2008, we had the first mutation-selective agent developed. So that was no less than about 6 years later. Of course, the world of kinase chemistry has been pretty well developed, so I think there was a lot of resources and reagents within the actual screening. By 2011, this is a New England Journal of Medicine article, and this is a patient with widespread metastasis who was given a selective BRAF inhibitor, in this case vemurafenib, and showing that, in fact, all the internal metastasis had essentially melted away. So by 2011, which is 9 years after BRAF was actually initially even observed, we had a target that was put under testing in trial that showed a tremendous response. And selective, again, for the BRAF mutation. So these are three of the pivotal articles that eventually led to the approval of the BRAF inhibitor and MEK inhibitor. So I'll just quickly summarize some of this data. In terms of BRAF inhibitors, there are two currently, vemurafenib, dabrafenib. This is called a waterfall plot showing the tumor response to the drug. And as you can see, most tumors show stabilization, some shrinkage by partial response, or complete disappearance as complete response. And they show relatively similar profiles, as expected, since they're the same target. If you look at progression-free survival, the length of time before a new spot appears, we see that overall, you can expect stabilization of disease or shrinkage until about, on average, 6 months, in which case they start relapsing. And if you combine both of them, we see that you can improve the actual progression by about 3 months. So unfortunately, most still relapse. This, of course, is sort of the overall survival on the two sets that we have, dabrafenib or trametinib, which is BRAF plus MEK, and vemurafenib, and here clearly showing that the combination of targeting both BRAF and MEK is superior to BRAF alone. So about 80% of the patients are alive on dual treatment at 1 year, and this is in contrast to about 5-10 years ago, where you're looking at average survival is about 3 months. So we've made tremendous progress, and we know that the combination reduces the risk of death and risk of progression compared to single agent. So this is sort of what I went through in a very quick way, sort of early phase, mature phase, FDA approval and clinical use, and now we have those drugs in clinical use. What I'm talking about really is sort of target practice, where we have some lead compounds maybe, or some compounds already in existence, and we want to sort of test ideas in a preclinical setting. I'm not an oncologist. I'm unable to actually give the drugs, but we'll talk about some of these lead targets. So when you start looking at this panel, there's a lot of areas where you can introduce new targeting. BRAF, for instance, can move around to all sorts of other molecules that potentially can be targeted for melanoma therapeutics. What I'm going to do is really talk about one that we're working at, which is rescuing p53, hoping to induce apoptosis and inhibiting proliferation by targeting the other MAP kinase pathways. So what is the rationale for this? If you actually look at the genetic profile of melanomas, about 40% of them show a BRAF mutation and a wild-type p53. So the idea is perhaps you can repress MAP kinase signaling while rescuing p53. So how does that happen? Well, in melanomas, as it turns out, most of the p53 is locked up by MDM2 or HDM2 in humans, ubiquitinated and destroyed. The reason being is p14ARF is frequently deleted in melanoma, and therefore HDM2 is allowed to run around wildly. So the idea is to interrupt that interaction with the MDM2 antagonist and release p53. So Nutlin-3 is the tool compound that we've been using, although there are newer generation MDM2 antagonists and multi-MDM antagonists, and it fits nicely into the MDM2 pocket. So in our first set of studies, we show, in fact, that the drug does induce p53 and its target B21, and in the mutant p53 cells, there's no such stimulation. So we know, and this has been confirmed over and over again, this was to design the initial drug screen, that the drug induces p53 and its targets. If you look at survival fraction, we see that, in fact, Nutlin-3 is much more effective in p53 wild-type lines compared to mutated lines, which is what you would expect if this drug truly was targeting the correct molecule inside the cells. So when you combine the agents, is there any sort of sense or element of interference? We show that MAP kinase suppression, in fact, and p53 rescue can be effectively combined, independently eliciting their effects. And you can see, in fact, wherever there's nutlins, you have the P21. You don't have it with a MEK inhibitor, which is U0126. If you combine the treatments and you look at its effects on the cell, we see that MEK inhibitors alone tend to be very cytostatic. However, if you combine MEK with nutlin-3, you induce a significantly greater amount of apoptosis, the subG1 fraction, confirmed by caspase activity. These are synergy plots. There are certain ways that you define synergy in terms of antagonism and in terms of synergism. And as you can show, the combination in this situation here shows that it's a little bit more than you would expect as additive, and that is how you define synergy. However, if you look at cells that are highly sensitive to Plex starting out, you don't get much synergy because by definition, they're already sensitive to the drug. So synergy works well either in low dose, where you have less toxicity, or in cell lines that are more resistant. We mapped out, again, in about 40 cell lines, the combination index, which shows antagonism here. If it's greater than 1, and tremendous synergy is less than 0.1, and we've color-coded this in a heat map way, and what we see is that a lot of cell lines show significant synergy across the entire dosing structure, or at least in certain pockets of opportunity. So clearly the combination will work. And if you look at a dual sort of genotype method, you'll see that the combination genotype predicts response to both treatments. So how about a BRAF inhibitor and NT3? You can't assume that these things are identical, especially if you want to bring the combination to drug treatment. So we pretty much did a similar set of studies looking at vemurafenib and NT3, and also showed concurrent inhibition BRAF and MDM2, a similar plot line of the combination indices as a heat map, and there's obviously, evidence is strong to very strong, shown in the green here, synergism between nutlin-3 and vemurafenib. And again, if you look at the effects of VEM on cell lines, it's largely cytostatic, G1 arrest. And this has been seen before, but if you're able to combine it with nutlin-3, you get more death. And this is shown in the live dead cell assay, where you can see a lot of green cells in DMSO. Less cells in the VEM, but not a lot of red. And if you were to look at the combination, you'd see a sea of red cells. So we know that it's much more lethal when you combine the two, as opposed to just having one or the other. And these are the obligatory mice studies showing that the combination in sort of lower doses of the drug in conjunction does show a tremendous effect in tumor growth. So how does it work? Well, this is one question that we wanted to address. Most people study combinations, but they don't think about how synergism actually is affected at a molecular level. So we profiled a lot of VEM response genes, and in fact, we found one of the ones that went down most highly was survivin, or BRCA5, which we also knew was a negative target of p53. So this allowed us to set up some experiments. First, sorry, that's not FA2, which I'll talk about a bit, but that's survivin, leading to induction of apoptosis. If we actually look at p53 deficient cells, we can certainly see that, in fact, the cells are still responsive to VEM in terms of driving down survivin, and p53 depletion abrogates the nutlin-3 effects, as you would expect. So we did the counter study, in which we went ahead and did resistance to VEM and seeing if nutlin-3 can work in that context. So we just showed that in p53 deficient cells, VEM can still work. So we had a cell line that was bred into resistance with the BRAF splice product, and we saw, in fact, survivin could go down in the native cells, but overall, the VEM resistance had no effect on p53 induction or survival suppression, but nutlin-3. So we know that the two pathways, even if you develop resistance to the other one, can still be functional overall. So what does this suggest to us? It suggests that there are sort of two separate ways that you can achieve synergism. For instance, down here, if you add vemurafenib, you see a drop in cyclin D, but no effect by nutlin-3. While here, you have an induction of p21, but no effect by vemurafenib. So they're independent effects, and they work together to suppress the cells. So we call this a collaborative mode of synergism. But there are genes like survivin, as I showed, that have independent regulation by both pathways, because you can create resistance in one and show effect in the other. And in this situation, you see that both VEM and NT3 here, nutlin-3, could suppress survivin. So we call this a convergent mode of synergism. And overall, when we start studying these combinations, we know that synergy can result from collaborative and convergent modes of interaction. So we know repress and rescue takes advantage of the high rate of BRAF mutations and the high rate of wild-type p53 status in melanomas. The targeted cocktails hopefully throw more of a cytotoxic switch, because with the large amount of relapse, it's most likely that your cocktails will have to kill as many cells up front as possible so you don't end up selecting for secondary resistance. The mechanisms of synergy, whenever you start doing cocktails, need to be studied, and actually that's not a huge area of analysis at the current time. Complex genotyping needs to occur, and that obviously stratifies your patients into smaller and smaller pools. And at the Mass General, Keith Flaherty is in fact leading up some human trials currently looking at the combination of MDM inhibition and MAP kinase. So in the remainder couple of minutes, I'm going to take you to another area that we've been practicing in, moving away from p53 and talking about a receptor tyrosine kinase called FA2, and that was foreshadowed in a slightly earlier slide. So FA2 is a receptor tyrosine kinase. I know Spiros is in the audience somewhere, and he's excited about this gene. And it actually has a kinase domain, like most tyrosine kinase do in the intracellular domain, and it's one of the largest families of RTKs. If you actually look at its interaction, the ligand ephrin A, or any of the ephrin ligands, are actually on the membrane. So this is a cell-cell interaction in which the ligand is cellular and the receptor is also cellular. So there's two components to it. There's a forward signaling through FA2 and a reverse signaling through ephrin. And what we do know is that the ephrin ligand will actually shut down signaling. So it's a sort of inhibitory signal. However, you take away the ligand and you overexpress FA2, it feeds into a proliferation pathway in cancer. So there are ligand-independent oncogenic effects for this receptor tyrosine kinase. So this is Elena Pasquale's figure from her website looking at F receptors that's been described in all of the TCGA data sets. And we can see that melanoma, in fact, has a high level of mutations in FA2, in the F receptors, including FA2. But again, this is a compositive of a lot of the F receptors. We got interested in this a couple of years ago because we found that FA2 is, in fact, overexpressed in a reasonable subset of melanomas compared to their normal counterparts, which are melanocytes. So when we looked at this more seriously, when you knock down FA2, there's an induction of apoptosis and a tremendous loss of cell viability. And you can see this in sort of 3D cultures when you take out FA2. And finally, if you genetically deplete FA2, again, with short hairpins, the tumors essentially collapse in xenograft implantation. So we do know that this is a viable target in at least a subset of melanomas. So when Durga Uda Kumar left, another postdoc came in and started asking the question, is FA2 survival involved in resistance to vemurafenib? So we bred about 10 cell lines into VEM resistance. This is done in vitro. And in fact, we saw a good number of them show upregulation of FA2 and activation of FA2. So certainly, it's one of the RTKs, and as you know, receptor tyrosine kinases have been implicated in resistance mechanisms. If you look at clinical tumor specimens, you also see that FA2 levels are increased in the post-relapse specimens. And in this case, these are patients on a combination BRAF and MEK. And if you stain the tumors as shown on the right, you see the increase in the amount of FA2. So on a correlative level, we know that it is associated certainly with resistance. So when you look at the cells and you overexpress FA2, what do you find? You find that there is in fact resistance to vemurafenib, and you do the counter experiment in which you deplete FA2, and you find that it restores sensitivity. That suggests that FA2 in fact is mediating the phenotype of resistance that you see. The other is if we looked at migration and you took out FA2, in the resistant cells there's a tremendous drop-off, but in the sensitive cells that don't appear to depend on FA2, you don't have that much of an effect, as shown in the figure there. And if you look at colony formation, the same sort of phenomenon occurs. All right, in the resistant cells, there was a greater effect of taking out FA2 than in the sensitive cells where you don't have as much FA2 around. So what is that saying? It's saying that resistant cells are more addicted to FA2 compared to their sensitive counterparts. So that suggests to us maybe we can sort of drug FA2 as a potential opportunity in the resistant population, or even preemptively to try to abort that. So we got together with our kinase guru, Nat Gray at the Dana-Farber. He's sort of our new kinase sugar daddy, certainly in town. He had developed a series of FA2 inhibitors through his large-scale medicinal screening, and these FA2 inhibitors were quite potent in the nanomolar sort of range, and you can see the map downstairs as to where they map. None of these are incredibly specific. They are selective, though, for FA2. It hits mostly the tyrosine kinase sort of molecules. And if you look intracellularly, you see the activation of FA2 here is diminished without much of an effect on protein levels, suggesting that in the cells at least is hitting some target of activation for FA2. If you look at sensitivity in terms of BRAF lines, NRAS lines, and even sort of double wild type lines, they all show pretty good sensitivity in the 100 to 600 nanomolar range. You kind of don't want to break beyond 1 micromolar if you're going to think about this in a therapeutic way. So if you take out FA2, it dampens the inhibitor response, suggesting that the molecule is the intracellular target. And again, if you look at sensitive versus resistant in terms of VEM, you find that the VEM-resistant cells are more sensitive to FA2 inhibition than its VEM-sensitive counterparts, supporting, again, the idea that VEM resistance is a shift towards VEM addiction away from BRAF addiction. If you look at apoptosis, it induces apoptotic response. If you look at synergy with BRAF and MEK inhibitors, again, we see a good amount of synergy between the BRAF inhibitors and the FA2 inhibitors. And if you look at the animal studies, we certainly see that the inhibitors shown up here in a A375 normal cell, both Plex and the FA2 inhibitor suppresses tumor growth. However, if you look at the resistant cells, you've completely lost its response to Plex in the resistant cell line, but you still get pretty good suppression with the FA2 inhibitor. What's sort of interesting, if you look at it carefully with this tunnel, again, FA2 inhibitors, even in the tumors, induce a greater amount of cytotoxicity in the VEM-resistant tumors, suggesting, again, for a third time, the idea that there's greater FA2 addiction when you see the VEM-resistant population. So, from our perspective, resistance can be viewed as an addiction shift from BRAF to RTKs, of which FA2 is one of them. There are other ones that have been described in the literature for sure. And FA2 upregulation during VEM resistance creates cells with higher invasiveness and metastases. And this was done also through collaboration with Kieran Smalley down in the Moffitt. New targets based on resistant mechanisms represents a new wave of potential drug development. But again, from my gut instinct, I think upfront cytotoxicity is still probably the best way to go. One of the problems is that although this is appealing, multiple resistance mechanisms could occur simultaneously, thereby making it difficult for you to chase all these independent sort of resistance mechanisms. So how do we view sort of the perfect marriage overall? If we look at all sort of molecular targets, and we look at all druggable targets that there are in fact compounds for, there's really only a pretty small sort of sweet spot, which is right in the intersection between the two. The TCGA for melanoma will be published if it's not already available online, and you will see a lot of evidence for potential new drivers within melanoma, and the trick is to kind of nudge that into that sweet spot so you can put it in a context of vulnerability. So in conclusion, in terms of target practice, I just want to say BRAF is a paradigmatic first-generation target. Next-gen strategies will involve multi-genotypes and will be much, much more precise. And again, this demands a cocktail approach, which then demands an understanding of how synergism actually works. Of course, people are talking about comprehensive approaches that will now involve combinations of immune and molecular therapies, because obviously one of the double-edged swords with this multi-cocktail is what is the effect of that on the immune system. You certainly don't want to harm one while trying to pursue the other. So with that, I just want to thank the SID again for the opportunity to speak in front of you. And again, it's been a wonderful weekend. Thank you.